thanks for coming out. And this is our sort of penultimate Water Ambassadors webinar. So let me share my screen here. Oh, I am sharing my screen. Can you guys see all that? So
yes, looks great, Vincent.
okay, great. So welcome everybody. And let me get over here. Why is this not? Okay, thanks everybody for coming out. This is our, if you're not familiar with Zoom, just to let you know, we have muted your microphone. And if you can't hear us, just click on that little arrow or caret and you can adjust your audio settings. And we do ask you to not start your video just because that does lead to bandwidth issues and trouble with transmission. So if you can leave your video off until after, maybe after the presentation. And you can see who's here by clicking on that participants icon there. And if you have any questions, please enter them into the chat. You can enter them anytime. We will get to the questions after the presentation. We'll sort of run down the list. But please, if you think of anything while the presentation is going on, feel free to enter it as soon as you think of it. And then that leave meeting button will get you out of the webinar. And then you can change just your view settings. Looking at that view options menu there, you can change the how we look to use, if that helps. We're also going to record this, so if you miss this or want to share it with your friends, please, or see it again, you can find it at this web link here. And I'll try to get that posted into the chat as we go along. Let's see. All right. And I don't know, are we recording right now, Lisa?
Yeah.
No. Okay, we are. Sorry. So again, as I mentioned, please use the chat feature to ask your questions. Again, as I mentioned, today we're covering clam restoration by Dr. Todd Osborne, who's at the Whitney Marine Laboratory at UF. Next, 2 weeks from now, sorry, August 24th, is our rescheduled webinar on the Gentian Beach Mangrove Restoration Project by Zach Hughes from Martin County. And also stay tuned. We'll have a graduates field trip. And right now that's looking like August 26th. So you may get an email about that. So just keep that in mind. So with that, our next speaker is Dr. Todd Osborne. And Dr. Osborne is Associate Professor of Coastal Biogeochemistry at the Whitney UF Whitney Lab for Marine Biosciences. Talk about the Indian River Lagoon and the impacts of the Indian River Lagoon. The work he's doing is one of those important solutions to kind of help with the water quality problems that we've had over the years, and particularly in recent years. And with everything going on with seagrass losses, manatee deaths, one of the things that you may not know is back in the probably late 70s, early to mid 80s, we had a very thriving hard clam population in the Indian River Lagoon, supporting commercial and recreational fisheries. And we've lost that. And the recognition of that over the last several years has led to the project that Todd is going to talk about. He's a biogeochemist by training, particularly interested in topics like carbon sequestration, mangrove migration, and coastal eutrophication, both locally and globally. And and he's looking into all kinds of things that can help improve the water quality of the Indian River Lagoon and other areas that he's doing his work. He has a background in degrees in biology, environmental engineering, and biogeochemistry. And he really works to combine his scientific interests and training with a lifelong passion for the environment. So with that, I'm happy to and very pleased to introduce Dr. Todd Osborne. And I'm going to stop sharing my screen, Todd, and I'll let you take it away.
All right. Okay, doke. Let's see if I can get this one right. We practice this. It's always the one, you know. I'm always the one that does that. Okay, let's see. You guys got a talk. All right, good.
There we go.
Okay.
Looks great.
Looks great.
All right, thank you. Well, thank you, Vincent. Thank you, Lisa, for the invitation. I see lots of names here that I recognize. Lorraine's here, Kent's here, Irene's here, folks that I work with, and my mother-in-law's here. Hi, Marianne. So the pressure's really on now. I better do a good job. Okay, so thank you for the opportunity to speak with you guys today. I know a lot of you are local to the Lagoon, and so this has special bearing for you, as it does for me. But our goal and what I want to tell you about today is a project or an initiative that's been going on for a while. We're working hard as a group to restore clam populations in the lagoon, and this is not by any means the solution to what's going on out there, but it's one of many approaches that we're trying to take to improve lagoon water quality and in the end get our lagoon healthy again. And so I want to acknowledge the folks that are working very closely with me. Of course, Jose Nunez, who is my clam whisperer and also aquaculture specialist. He does all of the spawning and rearing here at the lab, and he's a very special part of this project. Mr. Jeff Beal, who many of you are familiar with, who is now at Ducks Unlimited, was at FWC, and is still very active in this, and Lorraine down at FOS, and Vincent's old stomping grounds is very active as well. And then the rest of these folks, Anna, Sabrina, and Norm, are all parts of my team here at Whitney, and I'm very glad to have them helping as well, because it is a big endeavor. And I also want to say a big thank you to several groups out there that have been either helping us hands-on or helping us financially with this process. And we're in our third year of clam work in the IRL. And so Blair Wiggins and Addictive Fishing have been very, very big in helping us promote this. The Water Management District and their scientists are active with this project as well. We even have some corporate sponsors like Starbright, who through Blair Wiggins Group are helping us with this. And then so many agencies involved, FWC and NEP, the EELS program there in Brevard County, the One Lagoon group. I mean, all of these folks are working in the same direction. CCA and Keep Florida Fishing are big time as far as like really putting boots on the ground to help us. And I can't go anywhere without saying thank you to Mike Sullivan at Premium Seafood, who is my commercial partner, helping me grow out millions of clams every year in his facility, which is also here in St. Augustine, and many of his leases that are in the central and northern portions of the lagoon. So thank you to all of them. And I think I'm probably preaching to the choir here when I tell you a little bit about the IRL, but just for the sake of being fully backgrounding you guys, it's a big system, and I think you all can appreciate the impact it has. Probably one of the things that a lot of Floridians don't realize is just how many people live along the shorelines and within the 7 counties that border the IRL, and how impactful water quality in the lagoon can actually be. And it's not just an environmental issue, it's an economic issue as well. And I hope you captured that in my title when you saw it, that there's a lot going on with the lagoon, and the impacts are far reaching, in that while the system itself is really big, the impacts for declining water quality can be equally substantial. And that's not only to our natural estuary system and to our natural environment, but also to the human environment there as well. An estimated economic value in 2007 of $3.7 billion annually. I think that gets everybody's attention. From the scientific side of things, you know, these water bodies are extremely diverse as well, and have been recognized as such. You know, an estuary of national significance, and has been named the most biologically diverse estuary in North America, or was. And that's, there's so many different plants and animals utilizing this unique ecosystem. And so it's a very important part of Florida as well as the East Coast. And I think that, putting our efforts into trying to regain some control of the estuary and its environmental health will have broad reaching impact, all the way through to our economy. So it's a win-win, I think, for us to be doing this kind of work. Now, many of you probably remember a lagoon that looked a lot like this, clear waters, dominated by mangroves on the shallow and shoreline areas, and seagrasses in the benthos, and in most cases those seagrasses kind of forming the foundation of our lagoon system. Enter water quality problems, eutrophication, excessive nutrients, and we end up with algal blooms that in turn really cause major problems, and that can come in several different ways, right? Not only through harmful algal blooms that actually produce toxins, but also through the biological oxygen demand, or the lack of oxygen that's a result of all that algae, all those blooming algae dying back and consuming oxygen as they decompose. Now, we're aware of several different blooms that have been ongoing, and some of the information out there lets us kind of look at that spatially. But the north central Indian River, Banana River, and northern Indian River are some of the most hard hit places in the lagoon itself. And as you guys know, there's not a lot of flushing in the lagoon, with few inlets, very well spaced apart. The impact of tide and tidal flushing is very low in the lagoon. And so when things go south on us, and I mean that when things environmentally degrade within the lagoon, it takes a long time to kind of attenuate that signal, because there's not a lot of mixing with ocean water on a regular basis, right? And what you can see in these two figures, and these are older figures, but that they're kind of give us an indication of where those big problems are. One is a Corynea brevis bloom, and the other pyridinium, both species that can be harmful in the system. But on the norm, and I say that as things are going in the aquatic environment, and I say specifically in the saltwater environment, this is true in a lot of ways of freshwater, but to a much lower degree. But in our saltwater environments, we have filter feeders out there that are keeping algae in check. And there's two ways to look at this. And ecologists say that there's a bottom-up approach to that kind of control, and then there's a top-down. And the way this works is that a bottom-up control on population would be things like the resources that allow that population to grow. So bottom-up control of algae in the lagoon would be things like nutrients, nitrogen and phosphorus, and those types of things that end up making their way into the water column, and feeding kind of rapid growth of algae. So, the bottom-up controls on algae in the lagoon aren't really there right now, because the excess of nutrients that are available through our stormwater systems. Every time the rain comes in the rainy season, we see algal productivity increase dramatically in the lagoon, and it's a direct linkage to the runoff from our landscape. So not only our septic and sewer systems that are contributing nutrients, but also our fertilizer application to our yards and things like that. And there are a lot of programs, I want to mention it right now, there are a lot of programs in the lagoon area that are forward thinking and looking to reduce the use of fertilizers and switching from septic systems to sewer systems. These kinds of approaches are making headway, along with other things like muck removal and those kinds of approaches. They're all important ones. And so this is just one piece. But the other side of control of algae would be the top down. And what I mean by that is the control of population through predation. And in this case, it would be what we call herbivory, but eating those plants. And so what eats algae? Well, filter feeders do, right? So zooplankton, which are little tiny creatures in the water column themselves, will consume algae, and other organisms will consume those zooplankton, so they get into the food web that way. But then the larger macroorganisms, like oysters and clams and corals and things like that, are also filter feeders, and they're filtering out particles. Now they do two things. They filter out algae, which they consume, right? And cyanobacteria and those types of things that bloom in our lagoon. And they also filter out small particles that cause turbidity, which shade our seagrasses and cause us to lose them. So on both sides of that, there's really important activity in this biofiltration. We're going to call that an ecosystem service. You've probably heard that term before, and it's used to kind of describe the beneficial aspects, and from a human perspective, of course, from elements in the environment and what they do. And so filter feeders in the lagoon ecosystem provide this ecosystem service of filtration. And that filtration reduces turbidity, either mineral materials like organic matter or soil materials, fine clays and things like that may be suspended in the water column that create turbidity or shade. And then of course the live portions, the algae that they filter out and consume. And so as Vincent mentioned early on in his introduction, that clams used to be a big part of the IRL as well. And in fact, a lot of you probably remember, if you've been around in the last 20 years around the lagoon, that clamming was a huge industry here, and in its heyday was hyper productive in terms of clam harvest. But also people growing up around the lagoon remember just being able to jump out anywhere you are in your boat and fill a bucket with clams in no time at all. And so they were a substantial part of the environment. We're going to talk about what happened to them. But we say, well, why haven't clams, really done the trick for us? And I'll explain a little bit about that as we go. These are my friends, the Mercenaria, which we would call the northern quahog clam. For those of you familiar with clam chowder, the big guys, this is what you get. Usually northern quahogs, and their range is all the way up to Maine and down south of the lagoon. And they have a southern variety, we call it a species, but they do interbreed pretty easily, called Mercenaria campechiensis, and that's the southern quahog. And when they do hybridize, we end up with some pretty dramatically heat tolerant and fast growing clams, which is what aquaculture is really looking for in the South, right? So they're here and they've been here for quite a while, but they are reproductively viable when they hybridize, which is unlike other species. We almost think of these as genetic varieties, right? And one of the really unique things about reproduction in clams is that they're synchronous spawners. And what that means is that they spawn synchronously, at the same time, and that's their kind of reproductive strategy. And they do it at a kind of a large scale. Now, to get clams to spawn, it's usually a natural process in the spring and the fall. So we get 2 spawns a year when water temperatures change. And so the way to initiate spawning in the laboratory is we just cycle the temperature. When I say we just cycle the temperature, I think Jose turns the lights down and plays special music and things like that. But he really just cycles temperature. And so we will take the clams for a week or so at 70 degrees, and we'll drop them down to 60, and then we'll bring them back up to 68, and we'll drop them down to 62. And we cycle those back and forth in the laboratory over the course of a day. And sometime in the afternoon, the spawning occurs. And once that egg and sperm release happens, you can see it in our trays, and we collect all of that water and end up with lots and lots and lots of baby clams, right? So one thing to remember about these guys is a healthy adult clam can release 1 to 2 million eggs per clam. So in the environment, they're just seeding these out into the water column, and everything under the sun, of course, is eating them, but they have some level of reproductive success in large quantities, right? When the clams are close together in beds, they're really fantastic about being reproductively successful. Spread out and in small numbers, not so much. We'll keep that in mind because that's important for the story. The other thing that is so cool about clams is they do this thing called benthic coupling, and that's a really important factor as well from the biogeochemistry of this, and that is, how do clams really help us out? Well, they can only eat so much algae, right? But they continue to filter because that's how they breathe. And so that process brings in particles continuously. And while they may be full of consuming algae after a short time of filtering, the rest of that material still gets trapped on their gills and they still contain it. And what they do is they ingest that material out, they spit it back out. But now it's a particle. And I'm going to show you a video of that in just a second. The idea, though, is that those processes, both the filtration and consumption of algae, as well as the filtration and egestion out to the sediment surface, both result in the same thing, which is the reduction of particles in the water, and therefore clearer water that allows for the return of seagrass. And that's really where I think we're all thinking right now. We're all familiar with what we see in the news. Manatees are starving. It's a big crisis, and that all kind of comes back to turbidity in the water column. Shading of seagrasses don't grow. And for instance, if you were to take some sort of shade material, a carpet or a tarp or something, and put it over your yard for a while and come back a week or two later, you'll notice that you've killed your grass. And that's more or less an easy representation of what's happening in the lagoon when we have algal blooms. The light just doesn't get to the bottom. Seagrasses are photosynthetic, and so without that light, they can't grow and they end up dying. And that's the food source and the habitat source for so many organisms in the lagoon. So it's basically the foundation of the system, and that's what we want to get back. When we talk about ecosystem services, I can't say enough about the reduction of turbidity, but also there's this kind of retention of nutrients. And this little cartoon is actually from the UF IFAS shellfish extension group, Leslie Sturmer's group over at UF, and it's meant to illustrate to you not only do clams like retain those particles, but they also help in this process of reducing nitrogen and phosphorus. Both are elements that feed algal blooms in the system, right? And so, you know, with an adult Mercenaria clam filtering, not only takes in the organic matter, and so it actually stores some nitrogen and phosphorus in its body, but it also does this thing that you can see here on the screen, this biodeposits, where the stuff that is not consumed or not, you know, digested actually ends up being spit back out, but it's now a part of the sediment. And really great things from a biogeochemist perspective happen down there. You say, how much fun can it be in the mud? Well, it's actually really important. And so the same processes that we use to reduce nitrogen in wastewater treatment plants happens naturally out here in the sediments. And that is with the absence of oxygen around the, you know, right at the surface of the sediment and below, bacteria are working on this organic matter. They're releasing ammonia, which through a process is nitrified and then denitrified. And by that only means that, you know, it's turned into nitrate with an oxygen availability at the surface, and then bacteria right below the surface in the anaerobic zone are able to denitrify it. And so we take organic nitrogen and we turn it to nitrogen gas over here, which gases off. And that's the main mechanism of nitrogen removal. This benthic depositing process is what's so critical with clams. And oysters can do this too, which is really important. The other awesome part about that is they incorporate phosphorus into their shell, not only into their tissues, but into their shell. And I want to say that that's a really important process too. Now, one that we don't give credit for, at least not yet, is the fact that also these shells retain carbon as carbonate. Calcium carbonate is what makes up these shells, sometimes magnesium carbonates, but entrained in that is phosphorus, as well as lots of carbon, substantial amounts of carbon. And so these are also carbon sinks, which is something that's important to us. If you're hearing about our global warming issue, that's a big deal. And along with that is this issue of ocean acidification. You wonder what buffers our estuaries from becoming more acidic. Well, oyster and clamshell is the nature's version of Tums, right? It helps buffer the acidity that's coming in through stormwater and, you know, surface water inputs, as well as increased CO2 in the atmosphere. It helps push pH down. And in the marine environment, that's not good for a lot of organisms that create things, especially shell. So all of this additional material that's in the system has another ecological role as well, which is to buffer that pH. And we don't want to forget about that. that's a lot of chemistry, and you guys are all going, oh my gosh, enough, Dr. O, please. So I'm going to show you a little video about how clams filter feed and see if we can visualize this process that I think is so critical. Let's see. Okay, so couple of clams in a tank here in the lab. They do bury themselves. Someone asked me one time, can adult clams bury in the sand? Absolutely, and here's some proof. They do their thing, and then we add some algae. And you'll notice the picture of the seagrass in the back of the aquarium disappearing, and we can't see it. And then we give them some time to do what they do so well, which is filter this stuff out. And you'll notice they're pulling in water through their siphons. They have to do that to breathe. And at some point, they get kind of full. So now they're like, they've eaten enough, but now they're spitting out these aggregated clumps of algae. And look what's happened. Now the water's clear again. That algae that was in the water column, which would not normally settle out, is now in clumps on the bottom. And you can see these shrimp taking advantage of that. Because shrimp can't filter algae out of the water column, but they can sure eat clumps on the bottom, right? As can many other invertebrates in the, in the estuary. And so this benthic coupling is not only important for that process, creating or making that algae available to other consumers, it's also important for that nitrogen process that we talked about a minute ago in the diagram. I think that's super important. I did have a slide about the CO2 side of things, and I just want to mention that because we are working diligently to create a carbon market for marine aquaculture, such that this kinds of things can be captured and utilized from a farmer's perspective. And I want to make a pitch right now for the work of aquaculture in Florida, because not only is it an industry with economic impact, the techniques from aquaculture we are using on a regular basis here in this program, following those same techniques pioneered for aquaculture to, reinvigorate populations in the lagoon. And I'll talk more about that later, but I want to say that our industry of aquaculture in Florida can be very helpful to us in this process. So getting current farmers and new farmers involved in growing clams in the IRL and other places helps us in the long run by that top-down control on algae, as well as this whole idea of carbon storage and pH buffering in the system. All very important aspects. And if we do this right, teaming with the commercial producers, we can really have an impact. And that's part of our goal here in this program, is to include not just some scientists and restoration folks, but to get the bigger commercial producers of shellfish involved and interested in participating. And I think that's a big deal. Now, a couple of things that are also of great interest to us are, why clams and not just oysters? Because we have a pretty substantial oyster program already in place in the lagoon, doing great things. Marine Discovery Center and Brevard Zoo and many groups are working together to restore oysters, and it works. And it's Linda Walters down at UCF has been pioneering a lot of reef construction in the last 10 or 15 years. And so there's been a great effort to do that. And we think that it's because of that filtration capacity that we want to bring clams back into the picture too. I mean, a single 1-year-old clam filters about 10 gallons of water per day. Sometimes a little more, sometimes a little less, but that's on average. But that's kind of double the previous estimates that we've seen in the literature. We did this kind of work in my lab in a very controlled environment. But as you can see, they not only reduce chlorophyll, which is the blue dots there over time, they're also reducing turbidity at a very similar rate. And the idea there is that no matter what the cause of the shading for our seagrasses, if we can enlist the help of Mother Nature to reduce that, that's where we want to go. We want to get that turbidity down so seagrasses can come back. So why haven't the clam sticks problem already? Well, the biggest problem is that we took most of them away. And my colleague Blair Wiggins will tell you that he remembers as a professional fishing guide having to go to the boat ramps really early just to launch a boat, because all the clam boats were going to clog up those boat ramps in the morning, and they wouldn't be able to get out. And also that the fact that the industry for wild caught clams was somewhat, it's not unregulated, but it was a bit of a free-for-all there for a while, and there were a lot of a lot of harvesting going on that was documented, but also a lot of harvesting that probably wasn't. I mean, this is just one load of clams raked out of the lagoon. If you think about 400 to 500 boats doing this every day for 10 years, it's substantial. And the numbers look a little like this. These are estimates of pounds of harvested clams. Now we look at that. It's not clams with shells. We're talking about clam meat. That's a lot of clams. And peaks back in the early 80s and again in the early to mid 90s. Tremendous amounts of clam harvest. And this isn't on aquaculture leases like we do it now. This is kind of wild caught, fishery. So a lot of harvesting going on there. This green arrow is kind of the inception of our algae problems. Really started kicking in the late 2009, 10, 11. We had super blooms following there. But what we also see is that the harvest diminished tremendously because the numbers of clams just weren't there. And so whether it was overfishing or environmental drivers doesn't really matter. What does matter is when we see the loss of that biofiltration capacity, the result is the algal blooms that we've seen in the past. And this is not unique, sadly, to the IRL. There are other places that have seen this same kind of thing. This is clam harvest history of Great South Bay in New York. And you can see just tremendous, those are in thousands of bushels of clams, or it's amazing. 400,000, 500,000, peaking at 700,000 bushels, of clams in harvest. And what you see here is as the landings diminish, they diminish and they continue to diminish. Then we start seeing problems. The blue arrow here is the timeline for their harmful algal blooms up there. And so the trend, if we kind of toggle back and forth between these, is not so far off. And it's been documented up there, and now we're documenting it here. There is a strong relationship between the loss of filtration and these things. But so suffice to say, very important that we see that, or that we recognize that and move forward with it. And we can see here that, just looking at the loss of these organisms and what is going on now. On the left, you see chlorophyll during a bloom event, in 2012, and what those kinds of things looked like. Prior to that, we didn't see those types of heavy blooms. But then you'll also see on the far right a change in shell growth for oysters and their abundance, and add on to that this kind of large-scale loss of commercial Mercenaria harvest in local waters. These are all issues that we recognize coming together. Finally, though, of great importance here is that there was a tremendous loss of clams, but not a total loss of clams. And we're going to leverage that. And that's what we decided to do. Now, like I said, I'm a biogeochemist. I work on sediment and water quality and plant nutrients and things like that. What am I doing with clams? Well, the premise actually came from studying this kind of issue in a marine lab where all of my colleagues are molecular biologists. And by training, they are all very much interested in evolution. And so the idea came about one day over lunch. Well, if there is a few clams left in the IRL and we could find them, they'd probably be pretty tough, meaning they've survived years and years, onslaught of environmental insult. And they would probably be fairly stress resistant. And several people in the know down there kind of chuckled and said, well, good luck finding one. And I thought, no, that's not possible. So I spent quite a bit of time looking for one. And it took several hundred staff hours to come up with 39. And I say that kind of in a joking way, but also in an exasperated way, that it took a long time, several hundred staff hours of raking and looking in places that used to be high density clam beds to find a few single adults. And when we did, we were ecstatic. We actually ended up with 39 individuals after hundreds of hours of looking. And we took those 39 individuals home and fatten them up in the laboratory on scrumptious algae and then spawn them. And with those 39 clams, we ended up with 40 million larval clams. And it's a process. I'm going to walk you through that process because it's kind of fun. But these clams came mostly from the southern Mosquito Lagoon. And remember when I was talking about the synchronous spawning, like when the temperature fluctuations in the spring or in the fall trigger that spawning, naturally clams in big beds would spawn, and they would, all those eggs and sperm would get together and it'd be a successful spawning event. But these guys were, the closest clams we found to each other were probably 100 feet apart. And then that spanned over miles. Those 39 clams spanned over miles when we were collecting them. So they had absolutely zero chance of coming together. So reproductively, we have yet to find any small clams in the lagoon. However, good news is in this last year, we did find active veligers or larval clams in water samples around our site. So we know that they're reproducing now. Anyway, more about that later. The take home here is that they were not reproductively viable at all in the status that we found them. But bringing them back to the lab and spawning them in the lab gave us the opportunity to grow up their populations. And it's kind of cool to watch this in the lab and to watch the process grow. It takes, several weeks to go from freshly spawned fertilized eggs through the veliger stages, which are free swimming, but they look like little tiny clams. You can see the shell starting to develop and they're swimming around. And then when they set, when they're big enough and they're heavy enough, they fall out of the water column and they crawl around on the bottom of the tank, and they look for cover and they start developing their shell more so at that point. Now, oysters stick to things from their veliger stage. When they settle out, they find a place and they make it home. Clams are mobile, which is a benefit in some cases, but they're not mobile like fish. Of course, they can move around, but it's a slow process. Slow like my kids getting ready for school. But so the next step in that process is we grow them out in the laboratory. So as they're tiny little guys, we bring them up. We put them through our nursery system, and then we're filtering or we're pumping river water from the Matanzas River up here in St. Augustine, which is very clean, high quality water through the system, and they are eating algae there and growing. And you can see them on tanks, upwellers on the left and on the right, trays. And as they grow, when they get to be about 4 millimeters in size, which you see in the top left, then we can put them in bags and put them out in the environment and get them out of our raceways. And that's what you see here in the center top picture is Phil Cubbage, one of our local partners, preparing clam bags for field planting. So he takes all this small 3 to 5 millimeter seed clams. We put tens of thousands of them in a bag at a time, and then you can see them out in the river, tacked to the bottom. And they grow. And as they grow bigger, when they get up to about 10 to 15 millimeters, a little bit bigger than that, we can then replant them on the bottom under cover netting. And that cover netting allows us to protect them from herbivory. And that's what we're doing here. You can see on the left, we're prepping by pulling chlorpa from some areas. We don't want to tack that down underneath our nets, but we'll sow clams, and then we will cover them with what you see on the right here is a monofilament net that we remove after the first year. Because after the first year, post growth in St. Augustine ends up being more like 18 to 24 months in their life cycle. And then they're big. They're over an inch and a half in size. And that's when most commercial operations would harvest them and sell them to the fish market. We leave them in place, but we remove that cover netting gear so it does not become some sort of marine debris or fouling for other organisms. And at that point, they are less susceptible to predation. Lots of things eat clams, I'm sure you guys know, and their smaller stages, crabs and crown conch and moon snails are all predators. As they get bigger, once they get above that inch and a half margin, then it's only the bigger black drum and red drum and stingrays that can really consume them. And they do a great job of it. And we feed them pretty well with these projects because they can also get under nets and do things like that. But at that stage, the predation is minimized from our perspective. So we've grown them up in bags. We get them out there, and we harvest them from our leases. You can see some of our folks here, Adam Bassetti and Sabrina, working on that. And then Norm, you can see at River Rocks here in this other picture, planting them out. And I think this was originally a automated slide, and for some reason it's not, the animation is not working. But in the background is a map of sites that span from Southern Mosquito all the way down to Sebastian Inlet. We're working now with some partners in the Southern IRL down around FOS to grow out as well for this coming year. And the process is kind of labor intensive, but it's a fun one. We excavate those bags, blow all the silt out, take the shell out, and then repatriate them down to the IRL. And they look like this when we're planting them. So we're sowing them like Johnny Appleseed from buckets, and then we lay out our cover nets. And in the top right corner, you can see that happening. There's some COVID work going on in the bottom left. You can see us socially distancing with our masks. No fun there, but we are outside, and that was good. And then on the top right, you can barely see the net covering the recently planted clams, right? And the bottom right, you see Jose's somewhat patentable kite camera flying above, and you can see in these what I call a set is a 15-foot net, 50-foot long section. A big rectangle will put tens of thousands of clams underneath, ranging from 10 to 50,000 clams underneath one of those nets, depending on what's going on. But you can see them as things grow on the net, they start to get darker. And so from that picture, you can see those sets starting to become visible. If you look on Google Earth at some of our sites, you can see, we have lots of nets out there, and that they are growing lots of clams, and that's the goal, right? But so as a kind of a wrap up, our project status is like this. We have about 1.2 million clams in the Southern Mosquito Lagoon associated with some aquaculture leases, and we do that because they're protected that way. 1.8 million in Northern IRL, that's in the Lagoon area, in the Melbourne area, I'm sorry. And then about 5.5 million of various sizes down around Sebastian Grant area. And we've got a new site that we brought on board last year with the help of Sea Grant and some associations there, as well as the Florida Department of Ag and Consumer Services on 2 new leases in the Titusville area. And right now I have about 1.5 million there. If you were to fly over those sites, this is what you'd see if the water was clear. But each one of those squares down there, those sets, is a 15-foot by 50-foot net. And in fact, you can see the boat here from Google Earth, one of these processing days. And this is actually a picture of Turner Flats, I believe, which is in the Southern Mosquito Lagoon, right near JB's Fish Camp, for those of you familiar with the area. But this is a large scale, a 10-acre aquaculture lease owned by Mike Sullivan, and he donates the space to us to grow clams there. And in some cases, we actually will harvest some of these clams and move them farther south into more troubled waters. But this is kind of our stopover spot. We have a lot of plots established there, and the survival rate here is pretty tremendous. I mean, like 94% of what we bring from St. Augustine in this site is doing just fine. We still fight the blue crabs and everybody else and everything else that wants to eat clams. But as we move farther south, the problems change a little bit. Our survival rate goes down a touch. Not because of harmful algal blooms. In fact, last year's crazy algal bloom, we didn't notice any mortality associated with that or with the hypoxia that was associated with those blooms. However, lots of rainfall late last summer, which was actually 17 or 18 inches above normal in Brevard County, which already gets 50 some inches of rain a year, depressed the salinity quite a bit in that area. And so we noticed some mortality associated with salinity stress. That the Northern IRL is just chock full of crown conch, which love clams too. So that was another piece of that puzzle. But if you were to take all that we've done so far, which is close to 10 million clams repatriated over the last 3 years in the lagoon, we can estimate a daily filtration of about 100 million gallons. And you say, wow, that's a lot of water. Well, for a lagoon that size, it's not a lot. But we're getting there. We're starting to get to impactful size. And that's our goal, is to really tune this up and put this at such a scale that it's noticeable. Over 891 kilograms. That's like 2.2 pounds per kilogram, right? Almost a metric ton of nitrogen stored in clams, and that doesn't count for any of the benthic coupling that goes on. And about 300 kilograms of phosphorus stored. The surprising number here is how much carbon is being stored actually in clamshell from this project alone, which is roughly 27,000 metric tons, which is a lot, or 27 metric tons, sorry, 27,000 kilograms. That's a lot of carbon. But anyways, the take home there is that it does work, and that it's becoming impactful at the scale that we're working now. We've noticed some seagrass recruitment at some of our sites, which we're very happy about. And even after our first year, we're getting some public awareness too. And I can't say enough how much Blair Wiggins, who's in this picture, and Starbright, Danny over there on the left side, are helping get this message out to the public, and how impactful that is, and the help that we get, because awareness is really a part of the fight, right? And that's why I'm doing this talk today, and we've done several others like that. We want to get that word out to the public that this is another form of restoration that is doable, and we need your help to do it. So we've been on the TV a few times down there, especially with Blair's show. Lots of radio time with Catch a Memory and Captain Jim Ross out of Cocoa, and then lots of media coverage otherwise. I think the social media is where we seem to have a lot of impact. We've got 129 registered volunteers. Now that's not big compared to the oyster volunteer pool, but we're hoping to win some people over to get them involved in clam gardening as well. Lots of likes on social media, and to be honest, quite a bit of public donation to the cause too. So through groups like CCA and through Addictive Fishing Television, we've been able to capture quite a bit of giving from the public to help with us, and as well success through NEP and Brevard Tourism, and now DEP grants that are supporting this. And I think if you look back through the process, we've had a lot of growing internally as well. And I'd like to say that because several of the folks that have helped here, I know Irene and Kent and Vincent and Lorraine are all present. They've been involved in this, not only supporting through their agencies, but hands-on. So this is like an agency planting day where Blair came out with his television crew, and they actually created a show about this. But all of these agencies are represented here. FWC, St. Johns River Water Management District, and lots of our project collaborators were here for a planting day at River Rocks, which is super fun. But the take home that I want to send with you today is that spawning and rearing of this, what I would call rare broodstock. We're calling it the super clam because they are tough. They're tough as nails. These guys are really doing a great job, and they're very unique genetic line that we're taking care of here. This broodstock is successful and can be reliable for restoration activities, and that's a real important point to make. All of our sites are having survival overall above about 85%, but salinity incursions are still probably the most damaging to us. Some sites one site last year that we instituted, or early this spring, the Sampson Island project, which is a great project being done by Satellite Beach. Nick Sanzoni is the head of that, and instituting oysters, clams, mangroves, and seagrasses in one site. A pretty tremendous project, but the salinity dropped down to about 10 parts per thousand, which is a third of what it normally is, and kind of zapped our clams. So we're going to do another planting of clams in that area in hopes that they will take this fall. And we're thinking that full implementation before 2020 was our goal, and it was impactful then. Now in 2021 and moving forward, our goals are set higher, and we want to expand this to a much more significant degree. And conversations right now are about moving this into the Southern IRL with more significance, and expanding these populations in lots of small places. Working with a partner now, Brevard Zoo, our plan is to increase the distribution of clams and shorelines here with a new program called the Clam Gardener Program, where individuals with access to lagoon shoreline can host a planting of 10,000 clams in their own backyard, right? And maintain eyes on those sites. And an additional 100 small sites will be, I think, very impactful for the lagoon in terms of clam restoration, as well as natural repatriation through reproduction there. And I think that the take home for the broader scientific community, of course, is that we can leverage shellfish restoration just like we leverage plant restoration and other ecosystem service restoration techniques, and that we shouldn't ignore this one because it's so important. Our future plans, of course, to continue on. We want to engage commercial aquaculture to a greater degree, and we have some partners already developing to put these guys out on their leases and grow them. And we encourage them to grow them and harvest them and sell them, because that in one way is not only increasing the removal of nutrients, but it's also infusing that activity with some economy as well, and to try to give us some economic lift for the activities that are going on. If commercial aquaculture could take this over 100% in the future and be doing this at 100 million clams a year, we would be excited, and we could stop and they could take the torch. But we do think that continuing this process for the next 3 to 5 years is the right way to go, and we want to continue to engage the public with it, because your activities in this are super important. I encourage you to check out irlclamproject.com. We're updating that site now, but there's more information there, as well as the website at the Whitney Lab to learn more about it. If you want to get involved, contact us and we will put you to work. I want to say thank you for your time and patience. And if we've got a few minutes, Vincent, I'd love to have some questions. Thank you.
Thank you so much, Todd. There's a few questions in here, and I'll get started. I'd like to put our poll out, if you wouldn't mind, and if you please, for everybody attending, if you wouldn't mind filling that out while we'll go through the questions. Okay. So first question here from Bill Klein. He'd like to know details about the amount of clams and amount of clams and oysters in the IRL. I'm assuming that's historical populations. And then he asks, do you need any volunteer help in the Titusville area?
Bill, thank you for your question. Yes, absolutely. Contact me directly and I'll get you plugged into our team. In fact, I'll put my, let's see if I can jump in the chat, and I'll put my email in there so that you have that.
I pasted
pasted
a paper in there from Bill Arnold on clams, which I think will has some description of the clam fishery from what you had shown in your graphs in the 80s and 90s.
Perfect.
I have some resources on oysters too, but I just couldn't dig them up in this while you were speaking. So I'm going to put, Bill, I'm also going to put my email address in there too. So please feel free to reach out to me for some of that information. I do have some historical information or documents that talk about historical oyster populations. And there's one I'm trying to get, and while I get that, I will share that with you as well. Apparently there was some pretty extensive oyster beds in the main stem of the lagoon in Brevard. And there's a report from the 1970s that describes that. But I don't have a copy of that. There's someone I'm going to ask to get a copy of that. So, but the oyster populations of St. Lucie were documented to be around 450 acres before they in the St. Lucie River, not in the lagoon portion of our area in Martin County. But that was going back to the 1940s, I believe. Of course, it's not like that now. So the next question here is or comment. The great video from this is from Carson. Great video. Can we get a shareable link to that? Is that on your website?
Yeah. I believe it is on the Whitney website, but I will have to look and see. And if not, I will find that for you and we'll post it up. I think the narrated version is a little bit longer. Margie Lassie from St. Johns River Water Management District and Jose Nunez put that together here at the lab. Okay. They're husband and wife, by the way. And so they teamed up and made that video. And I like to show it because it just makes the visual is so clear of what's going on there. But the video itself will exist somewhere online if it doesn't already. And I'll have to send that to you, Vincent, and you can distribute to the groups. I do not have it at the tip of my finger right now.
Okay, great. Yeah, it's a Echoing that, Swarna comments is a wonderful video, and I thought so too. It's really well done. But anyway, that would be a good start for everybody here is maybe look on the Whitney Lab for Marine Biosciences website. Is that a good
Yes.
place to start? Yeah, okay.
Yes,
Great.
and I'll make sure we get that up there sometime today. The other thing I'd like to pitch is that Blair Wiggins' group, if you're familiar with him or you tune into his show, Addictive Fishing Television, they have done several videos with us. They've come up to the lab and kind of documented the spawning process, the grow out process, and a lot of discussions are available on some of their YouTube channel and stuff like that. So they've got if you want to see more detail on those aspects from a much better speaker than me, you can tune in to what he's got going on and check in with his website and see that, because they spend a lot of time and a lot of production effort in making some educational outreach videos that do document those processes with greater detail and much better visuals, of course.
Cool. Yeah, and I've seen some of those myself, and I can attest to that. They're very good. And then I think I posted the IRL Clam project in the chat there as well, which could be another good starting point. So next question from Marley's is what is FNU on the graph? I don't remember what graph that was.
See if I can cruise back and see. Not this one. Well, I'll have to remember myself. It may have been the turbidity graph, I believe. This one.
Yes.
So formazine nephelometric units. This is a unit of turbidity. And basically it's a chemical compound we use that turns water kind of a milky white, but it's a way of quantitatively describing the shading of light. And so this is that's actually the chemistry that we use to calibrate these instruments to read turbidity, right? So the formazine is a chemical compound. Nephelometry is just the measure of turbidity, right? So all that $10 word there just tells us it's how we measure turbidity, which is the yellow or the orange dots, and then chlorophyll is a subcomponent of that turbidity. And if you were to look at this graph more from my perspective, what this tells me is that there's the turbidity and the chlorophyll reduce at the same rate or almost identical rates. The interesting part about it is that you could also look at this and say, well, chlorophyll isn't all of the turbidity that's in the system, right? Within our laboratory, the gap between chlorophyll and the turbidity units that you see here represents other stuff. It could be algae, it could be mineral solids that are being suspended, like silt particles or clay particles or whatever. And so while chlorophyll is one way of measuring it, that actual turbidity includes a lot of other things. And that's what you see in this figure, is that they're both reducing in concentration, but chlorophyll is usually what we track. Turbidity can be higher, and so other things can contribute to decline of seagrasses in the lagoon, not just the algae, which is the #1 contributor, right, but other things too. So containing our silt from leaving stormwater inputs and things like that is also important.
Cool. We're getting close to 1 o'clock, but I'll just go ahead and go through this if that's okay with you, Todd. If you got
Sure.
a couple of minutes.
Mhm.
So at 1 o'clock, if anybody needs to leave, feel free to leave. I'll go a few minutes over so we can cover some of these questions. Lorraine asks, what caused the dip and rebound of clam pounds harvested in the early 1990s?
That's a good question. And I wish Bill Arnold was here because he could probably tell us. And I'm working with Lorraine and Jeff Beal and Cassandra Armstrong from South Florida Water Management District to do a clam restoration workshop. And this is originally scheduled for September. We're a little bit worried about COVID right now, but this is one of the topics of discussion that we have planned for that workshop. And we do have Bill Arnold slated to speak at that. And he has probably the most recognizable person to have their finger on the pulse of clam population dynamics in the Indian River Lagoon and basically built a career on that bivalve ecology in the system. I've not asked him about that. I believe that there are several drivers for clam population. One of course is environmental, and then the other aspect, of course, is the natural variation in population dynamics, and so that could come from the weather. Too much fresh water, maybe a hurricane or two, putting a lot of fresh water in the lagoon could change those populations. So I'm not sure, Lorraine, what's driving that, but that's a good question and one that we should find out when we get a chance to talk to Bill.
Cool. Yeah, Bill he's the man when it comes to all that. And maybe that paper I posted in the chat will mention that as well. I haven't looked at that in a while, but there might be some information about that in there as well. So Carson asks, how many gallons filtered with this project? So
cumulatively, wow. I've got to calculate that. Daily, about 100 million, which is great. If we look at it across time, I think I did calculate that at one point for a report, and we're looking at billions of gallons. But over 3 years, it's still a drop in the bucket. And I'm not gonna say that what's happening here is not substantial. It is, but in the scale of the lagoon itself, we need to double this every year for a couple of years, and the natural recruitment will follow suit as we get enough out there that are reproducing, and when we model that, we kind of think of that each clam will at least reproduce itself once, right? So doubling of population is what we think of, and so 1 out of a million clam eggs surviving is enough to double that population. And clams can live to 10 to 15 years. So we're looking toward that as being a part of the model, and that as this goes forward, those populations doubling each year, it's a pretty impressive growth curve. How much water so far, I don't remember exactly, but I think it was somewhere around 3 billion gallons, something like that. And that grows every year. So like I said, our goal is to really be impactful, and to do that, we think a 3 to 5 year continued effort with help of Mother Nature is the way to go. Hopefully we can do that with your help.
Yeah. Cool. Yeah, it's a marathon, not a sprint. So next question, you got some great comments here. But our next question from Sue is, how do you decide where to place the clam beds in the IRL, avoiding boat anchors, freshwater runoff spots, seagrass beds, oyster beds, etc.?
Yes, all of that. And then there's some other tactics involved as well. So we started out only using aquaculture leases, because FDAC said, well, if you guys are going to culture these things, you're going to have to put them back in an aquaculture manner, because you are kind of following that protocol. And so that was very beneficial to us to use aquaculture leases from partners or donations, or even from FDACs themselves, letting us into some unclaimed leases in some areas. But those rules have changed recently, which is great. For the purpose of restoration, the rules around using leases have been relaxed quite a bit. And that means that we can start doing some native bottom planting in other areas, like the Clam Gardener program. We're just going to have 100 small sites in people's personal shorelines that they can actually go out and maintain. But major considerations, of course, is we will do no harm, meaning if there's an existing seagrass bed or something of that nature, we're not going to mess with that to plant clams. We'll find another place. We do find that co-planting with areas that either have oyster shell or active oyster bars is a great tactic, because clams like to burrow down in there, and that's like their own natural version of a cover net. And we do find that being close to stormwater outfalls is a big negative. So we try to avoid the larger freshwater inputs, because they do get so low. Physiologically, clams just can't survive below 15 to 16 parts per thousand saltwater for more than a week or so. It's just not feasible. No matter how super they are. That's something that's just a physiological limit. And so we are very cognizant of freshwater inputs there. So we try to look for things that will not stress them. And we also try to plant them in places where we get maximized oxygen and minimized predatory stress. So sometimes that's shallower areas. We tend to focus on the western shore, but we found a lot of really good sites on the eastern shoreline as well that we're using now. And as things open up, I think we'll utilize that more.
Yeah, those new laws or regulations are kind of hot off the press. That really just came out this past year, I believe. So, but yes, it definitely has opened things up for restoration. Next question: are there any prior successful ecosystem scale clam restoration projects you're aware of?
Now, I know that we're working in parallel with colleagues on the Gulf Coast, the Gulf Shellfish Institute, and partner groups over in Tampa Bay and Charlotte Harbor and areas like that are following the same kind of protocol. I think they're using a more direct association with commercial aquaculture and utilizing clams that either survived red tides and or excess production by aquaculture that can't necessarily make it to the aquaculture market. We've dabbled in that ourselves with Sea Grant, a very successful project in Titusville as COVID relief. But in general, I think that's it. I think what's going on in the IRL is very novel, and we feel like that was a good, interesting thing for us to jump into. And the Gulf Coast group in Florida is, I think we're on the same track, and we're communicating quite a bit to push both of ourselves forward. That's kind of the goal of that workshop I mentioned in the fall here, was to get all of us together and kind of push this agenda even farther, and what we know and what we can find out to maximize the project potential. But as far as restoration work with clams, this is it. Like I said, the East Coast contingency and the West Coast contingency, and I don't think it's happened anywhere else in the world yet. So this is a relatively new direction.
Yeah, and all in Florida too. So that's great. Last question from Sue again. Do you anticipate the spawn of the super clams to retain their parents' hardiness, or will they weaken over the generations?
Well, there's a lot that goes into genetic diversity and variability in your offspring. And so when we look at that from the scientific side, yes, there will be some dilution of that, as well as the natural recruitment of other genetics in the system. And so I think that will over time change, but those traits will be retained in a substantial portion of those offspring. And so, for the time being, that's a good thing. As the process goes forward and the lagoon becomes more healthy, those traits won't necessarily be required. However, they'll be retained in the greater diversity of the system. And we encourage genetic diversity and drift into the system so that that the things go forward naturally. And we think we'll get that as conditions improve. So as that kind of goes forward in time, in 10 years, you probably won't see dominance of one variety over another. And that's where we want to be. I mean, that would signify to me that we've done the job. But for the time being, I think we're going to see those traits naturally selected for until the lagoon is actually like healthy again. And we're not there yet, but I see that as being a potential in the future.
Cool. Thanks, Todd. And so that was the last question. There's some great comments here in the chat, and there's some interest for volunteering. For those people, if you can refer. Todd just posted his email in the chat. I did as well. So if you have any questions for both of us, you can email us. Kent Smith, sounds like Kent wants to give you some money. So
I know, I saw that, Kent, and yes, I'm ready to talk. Thanks, man.
All right, and with that, Robin ends looking forward to the sequel, Clam NATO. All right, well, there you got a project title right there, Todd. So.
All right.
All right, and thanks everybody. Thanks for filling out the evaluation. Thank you for coming. And remember, August 24th is our last Water Ambassador webinar for the season. So hope to see you. Also ending on I think a good note with regards to habitat restoration on a fairly large scale with mangrove restoration in Genting Beach. So anyway, thanks again, Todd, and thanks everybody for coming.
Thank you.
All right. Cool. Go ahead and pull. That should save it. All right.
All right, Vincent, have a great one, man. Thank you.
Yeah, thank you so much, Todd. That was awesome. Yeah, I'd love to talk to you more about the whole clam garden thing. That's you know, I think that's a great approach to get people to, you know, just adopt a small population under their dock, and you can be real strategic about site selection and finding ideal places to do it. And I think that would be a really good option for people, because not everybody wants to plant seagrass or put an oyster reef. It feels more final, you know, like I've had comments where people worry about, well, if I put this oyster reef here, what if I want to do something to my dock or put in a dock even. So, but clams kind of give you that extra flexibility, and while still doing some of the same things that we have tried to do in the past with oysters. So.
Well, the phone's ringing off the hook already. All right.
Yeah,
We'll
there you
talk
go.
some more, Vincent, but let's get together and do that.
Okay, cool. All right. Take care.
All right. Take it easy, man. Thanks a lot for the opportunity.
Yeah, thank you. Bye-bye, everybody.